आवाज नहीं करना, खाती आवाज नहीं करना, चालू कर देता। नमस्कार। टाइम हो गया ना, सबको बोलो, दूसरों को भी बोलो, स्टार्ट हो गया। हेलो। हेलो हेलो, आवाज आ रही?
हाँ सर, आ रही आवाज।
हाँ ठीक, ओके। हाँ, दूसरे पार्टिसिपेंट को भी बोलो। हाँ अगर आप ठीक है, स्टार्ट करेंगे।
हाँ सर, हम देखते।
हाँ okay. Today we are going to see Kochs postulates. Actually, postulates means the views, and here the Koch is name. Postulate means view, and Koch is a name of the scientist who proposed these postulates. So here this is the Koch postulates. Okay, see here. Attribute to robot Koch. Actually, the name of the Koch full name is robot Koch. He is a German scientist born in 1843. He is considered by many to be the founder of bacteriology. He is considered as a founder of bacteriology. Actually, he works on bacteria, and he born in 1843 as the district medical officer. One of the Koch's primary concern was the prevalent occurrence of anthrax among farms animal in
सर
the.
स्क्रीन नजर नहीं आ रही।
इसकी नहीं आ रही?
नहीं आ रही सर, सिर्फ गूगल नजर आ रहा।
अभी
नहीं सर।
अब आ रही क्या?
नहीं सर। हाँ अब आ रही।
क्या दिख रहा है?
माइक्रोब्स एंड ह्यूमंस।
अभी
हाउ डू वी नो दैट, वो वाला।
ओके, सी है। हाँ देखो, स्टार्ट करता हूँ। यहाँ से स्टार्ट रखना। कोच postulates. कोच बोले तो एक्चुअली नेम ऑफ साइंटिस्ट रोबोट कोच। postulates बोले तो व्यू, उसके व्यूज रिगार्डिंग द पैथोजेनेसिटी। याद रखो, रिगार्डिंग द पैथोजेनेसिटी। नेक्स्ट स्लाइड। रोबोट कोच is the full name. Robert Koch. He is a German scientist born in 1843. He is considered by many to be the founder of bacteriology. Most of the people, they consider that this Robert Koch is a bacteriology, founder of bacteriology, and he was born in German. Actually, he was district medical officer, and he discovered the anthrax disease among the animals in his area. After converting his 4 room into his own medical laboratory, he convert his house into medical laboratory with one microscope, with one microscope, and in this manner he set a laboratory in his work. So actually, it is a German scientist. He is founder of bacteriology. He was medical officer, and he convert his house, 4 room house, in to the laboratory with a microscope. Next slide, see here. This is the robot home. Actually, He was born in 1843 and he was died in 1910. German scientists formulated the bacteriological techniques, staining methods, discovered the Mycobacterium and Vibrio cholera. Okay, this is about the brief about the microbes and humans. See here, actually microbes actually that means the microorganisms. Here, these are the three concepts. One is very few microbes are always pathogen, always pathogenic. It means they are very few are pathogenic. Many microbes are potentially pathogenic. And third is most of the microbes are never pathogenic. Okay, these are the three things regarding the microbes or microorganisms. Very few microbes are always pathogenic. Many microbes are potentially pathogenic. Most of the microbes are never pathogenic. Next year, huh? How do we can how we do know that a given pathogen causes a specific disease? We see here, this is the diagram. Potentially pathogen isolated from or detected in clinical sampling. Actually, this is the sample of pathogen. This pathogen, when enters in the patient, it shows the symptoms. Here, recognize syndromes. Example, septicemia, endocarditis. These are the symptoms. Here, the diagnosis and effective treatment of infection depend not on just on isolation in organism, but in establishing a plausible link between the laboratory findings regarding syndromes and the patient clinic condition. Okay. Next, let's see here. How do we know that a given pathogen causes a specific disease? That is known as Kochs postulate, or how we can know that a given pathogen causes a specific disease. For example, Plasmodium causes the malaria. How we can know that Plasmodium causes the malaria disease? In the same manner, first this actually postulate produced by the coach, the pathogen must be present in the every case of the disease. This is the first thing. The pathogen, actually that is the disease causing agent, must be present in the every case of the disease. The pathogen is associated with the disease. The second thing, the pathogen must be isolated from the disease host and grow in a pure culture. In first step, the pathogen is associated with the disease or host, and it is isolated means it is separated from the host and grow in a pure culture. Grow in a pure culture. The specific disease must be reproduces when a pure culture of the pathogen is inoculated into a healthy susceptible host. It means that this pure culture of pathogen is inoculated or transferred into a healthy susceptible host. The pathogen must recoverable from the accidentally infected host. This pathogen shows the symptoms and it shows in host of previous disease. So these are the 4 points regarding this Kochs postulate. See here next slide. Robert Koch hypothesis. Koch hypothesis that anthrax bacillus, a gram-positive bacterium, was the cause of anthrax disease. The second thing, the Koch proves his hypothesis corrected by infecting mice with the bacillus strain taken from the spleen of animals who died from the disease. The third thing, when the infected mice shows identical symptoms, Koch proved his hypothesis corrected. Koch then sought to prove that anthrax that had no prior contact with animals could cause the same disease when introduced to animal host. Koch grew the bacilli in fewer culture for several generations. He then showed that they could still cause anthrax in later generations. So here the robot shows that the pathogen, when they isolated from the infected person or from infected animals, when they separate, cultured, when they cultured in pure form, and when they transfer to the animal or as well as plant, when they shows that symptoms of that disease in healthy plants or in healthy animals or in healthy human beings, then the robot hypothesis is correct. Robert Koch perfected his methods of diagnostic and expanded on the work of others. Koch invented the method of cultivating bacteria on nutrient mediums using potato as a source of nutrients for bacteria and created a medium that could be stored in dishes created by his colleague Petri. The Koch works on disease and diagnosis culminated with the creation of what are now known as Koch postulates. Koch postulates are four steps necessary to confirm if the suspected pathogen is indeed the cause of disease. Robert Koch clarifies with hypothesis. First, prove that the bacteria caused disease. Anthrax broke out in the local cattle, found the agent Bacillus anthracis by an experimental process now known as Koch postulates. But see here, these are the steps. Actually, what is Koch postulate? See here, the microorganisms are isolated from the dead animals. The microorganisms, they are isolated or separated from the dead animals. These microorganisms are grown on pure culture, that is artificial culture, that may be nutrient agar or potato dextrose agar. Generally, the nutrients agar is used for the growth of bacteria and potato dextrose agar is used for the growth of fungi. So in the post postulates, these are the steps for the confirmation of pathogenicity of the particular pathogen, whether this pathogen causes particular disease or not. That is known as post postulates. In first step, the microorganisms are isolated from the dead animals. Microorganisms, they are isolated from the dead organisms. The second, the microorganisms are grown in fuel culture. In third step, microorganisms are identified, whether it is bacteria, whether it is fungi, whether bacteria, which type of bacteria, what is the name of that bacteria, when it is fungus, which type of fungus is there. In the third step, see here, microorganisms are injected into the healthy animals, into healthy animals or healthy plants. Next thing, the disease is reported in the second animal. If the disease is reported in the second animal, microorganisms are grown in fuel culture again. Identification of identical microorganism shows that the disease is caused by the previous microorganism which is isolated from the dead animals. Okay? Next thing, here this is the diagram. Here the coach confirm his postulates. Here again I have divided in this diagram into two separate diagram. See here, in the first step, this is the mice or rat. These microorganisms are isolated from the dead animals. From this, the microorganisms are isolated and they are grown in artificial media or nutrient agar or either it will be PDA. These microorganisms are grown in pure culture. They are grown in pure culture and they are identified. Okay, next happen. The microorganisms are injected in the healthy animals through the injection. The disease is reproduced in the second animal. Microorganisms are isolated from this animal. The disease is reproduced in the second animal. Microorganisms are isolated from this animal. So, in the healthy animals, again the pathogens are present. You can see here, this is the pathogenic microorganism are grown in a pure culture, and here identification of microorganism are identified. Identical microorganisms are identified. It means that in the dead animal and in the healthy animals, the pathogen they are same. That is the identical microorganism. Next thing, exception to Kochs postulate. See here, what are the exception to the Kochs postulate? Here, the microorganisms that are unable to be cultured on artificial media. For example, tree phonema pallidum. This is the one bacterium that does not grow on culture media. Two or more organisms work in synergy to cause a disease. Symptoms and disease can be caused by any one of the several microscopes. Here, understanding severity or duration of the disease. Not all diseases behave the same. The diseases vary widely in their onset, duration, and level of activity. Diseases classifiers. Acute diseases symptoms develop rapidly and tend to be more severe. Chronic disease develops slowly and are less severe. Subacute disease symptoms between acute and chronic. Latent disease with a period of no symptoms when causative agent is inactive. The host has the disease what has subclinically symptoms. Thank you. So this is about the Kochs postulate. So the single slide, see here, the single slide, actually slide number 10. This slide shows the Kochs postulates. Here you remember these steps. What the Kochs done or what is the Kochs postulate. In the first step, microorganisms are isolated from the dead animals or from the infected plants, from the infected animals, from the infected host. This is the first step. This is the second step. Microorganisms or pathogens are grown in a pure culture. That is what media, whether it may be nutrient agar or it may be potato destructs agar. Again, the second step, it is a second step. Microorganisms are identified. Yeah, this is the second step. First, the microorganisms or pathogens they are isolated from the host or infected host, whether it may be animal, plants, or dead animals. These are grown in pure culture and these are identified. In the third step, the microorganisms are injected into healthy animals for confirmation of pathogenicity test. The disease is reported in second animals. Means the disease is reported in healthy animals. The disease is reported in the healthy animals. The microorganisms are grown again in pure culture. In the this is in the second step, the identification of identical microorganisms If these microorganisms or pathogens are same as in healthy animals after introduced is similar to the dead animals, it confirms that this is the pathogen that is causes the diseases or particular pathogen is responsible for causing the particular disease. So this is the Kochs postulate. Only if you remember these things, it is sufficient because in examination it is asked for 5 marks. Kochs postulates for test of pathogenicity. From this, we can test or we can confirm any disease which is caused by pathogen or which pathogen causes the particular disease. For example, if we know that no, these days different new diseases they are developing. Through these postulates, we can confirm which type of pathogen is responsible for causing the which type of disease. So for example, if the malaria, which is caused by Plasmodium vivax, we can confirm this disease by using this Koch postulate. So in the same manner, new disease can be confirmed or new disease pathogen can be confirmed with this Koch postulate. So it is essential to know what is the Koch postulate, how it is important in plant pathology or animal pathology. So this is all about the Koch postulate. So in examination, the question will be asked for 5 marks. If you know what is Koch postulates, then you will very easily understand which pathogen is responsible for development of which disease. And again, this pathogen is introduced into new healthy animal or plants. Again, they show the same symptoms. And again, they are similar pathogen as they are present in previous host, as they are present in the new healthy host, then it is confirmed that this is the particular pathogen that causes the disease. Okay, so this is about the post postulate. Besides this, you remember who is Koch when he is born. Actually, what is his position? Actually, he born in 1843 and he was a medical officer, district health officer, and he born in Germany. He was founder of bacteriology. Actually, he works on bacteria. So this is about the post postulates. If you have any doubt, you can ask the question. आवाज आ रही ना? हेलो हेलो आवाज आ रही ना?
Yes, sir.
हाँ
Yes,
तो
sir.
हाँ तो पोस्ट postulate 5 मार्क्स का एग्जाम में हर टाइम आया वाला है. देखो हर क्वेश्चन में अगर आपके समझ में पोस्ट postulate आ रहा है, पोस्ट postulate एक्चुअली क्या है? ये एक जरासीम डिसीज कैसा पैदा करता है, अगर वो जरासीम है कंफर्म करने का है, अगर वो हो रहा है डिसीज, तो ये तरीका किसने बताया? पोस्ट postulate में. पहले क्या करते हैं?
Cost postulate.
कोच postulate. इसको कोच बोलते, इसको बोलते कोच postulate. एक्चुअली द कोच नाम है साइंटिस्ट का, रॉबर्ट कोच. और postulate बोले तो उसूल या नजरिया, उसके ख्यालात, उसको बोलेंगे postulate. ठीक है. उसने क्या किया? माइक्रो ऑर्गेनिज्म, वो क्या किया? माइक्रो ऑर्गेनिज्म बोलते पैथोजन जिससे डिसीज होता है, वो क्या किए? जो डिसीज है, फॉर एग्जाम्पल एक आदमी को समझो टाइफॉइड है, ठीक है, अभी मालूम नहीं था टाइफॉइड क्या है, तो उसने क्या किया? उसको उलट लिया, बराबर है, उलट ले क्या किया? उसके जो टाइफॉइड जिससे होता है उसको सेपरेट किया, ठीक है, प्योर किया, प्योर फॉर्म में कल्चर किया. उसके बाद क्या? उसको आइडेंटिफाइड किया कि भाई है क्या ये, whether it is bacteria, whether it is virus, whether it is other microorganism, whether it is fungi. और वो क्या किया? वो वायरस, वो बैक्टीरिया, वो पैथोजन उसने क्या किया? हेल्दी एनिमल में प्रोड्यूस किया, इंट्रोड्यूस किया. हेल्दी एनिमल में इंट्रोड्यूस करने से क्या हुआ? फिर से उसको डिसीज दिखा हुआ, कौन से हेल्दी एनिमल में। फिर से उसने क्या किया, वो ऑर्गेनिजम्स को फिर पैथोजन को प्योर कल्चर फॉर्म में निकाला। अगर समझो वो ऑर्गेनिज्म थे आइडेंटिकल वन, देन इट इज कंफर्म दैट दिस डिसीज इज कॉज्ड बाय दिस पर्टिकुलर पैथोजन। ठीक है, समझ आएगा पोस्ट पोस्टुलेट क्या है। हेलो, आ रहा ना, सुनिए, आवाज आ रही ना। हेलो,
यस सर।
आवाज है ना, ठीक समझ में आया पोस्ट पोस्टुलेट क्या है। ये क्या है,
हाँ सर।
पैथोजेनेसिटी कंफर्मेशन करना है कि वी पर्टिकुलर पैथोजन कॉजेज द पर्टिकुलर डिसीज। हाँ, ऐसा कौन सा डिसीज, कौन सा
यस
पैथोजन से
सर।
कौन सा डिसीज कैसे कंफर्म करना है, इससे अपन इससे कर सकते। इसमें इतना बताने का है। हाँ, इसमें
यस
अगर
सर।
स्क्रीन पे जो 5 स्टेप दिख रहे हैं ना, 5 स्टेप, अगर आप एग्जाम में इतना भी लिख रहे हैं तो आपको
हाँ
पूरे मार्क मिलेंगे। इतना, इतने स्टेप्स लिखना, याद रखने का। हाँ, पहले पहले स्टेप में
यस
क्या है,
सर।
माइक्रो ऑर्गेनिजम्स आर आइसोलेटेड फ्रॉम द डेड एनिमल्स।
माइक्रो ऑर्गेनिज्म आर आइसोलेटेड फ्रॉम डेड एनिमल्स।
हाँ, दूसरे स्टेप में क्या है, आइसोलेटेड बोले तो
माइक्रो ऑर्गेनिज्म आर ग्रोन इन प्योर कल्चर।
प्योर का, आइसोलेटेड बोलते सेपरेट करने का।
जरासीम।
हाँ, इसको क्या करना है फिर,
हाँ।
ग्रोन इन प्योर कल्चर। प्योर कल्चर बोले तो बैक्टीरिया का वास्ते कौन सा यूज करते हैं, न्यूट्रिएंट आकार। ठीक है, और फंजाई के वास्ते कौन सा ग्रोथ करते हैं, प्योर
यस सर।
कल्चर, पीडीए, पोटैटो डिस्ट्रिक्ट आकार।
हम्म।
अब तीसरे स्टेप में क्या कर, दूसरे वी में क्या करे, वो पहचानने का कि कौन से पैथोजन है। बराबर है,
यस सर।
कैसा पहचानेंगे वो, उसकी की रहती है पहचानने की। ठीक है,
माइक्रो।
हाँ। अब तीसरे स्टेप में क्या करेगा, जो पैथोजन हम पकड़े वाले, आइसोलेट किए वाले,
माइक्रो ऑर्गेनिजम्स इंजेक्टेड इनटू हेल्दी एनिमल्स।
इंजेक्टेड होता है उसमें वो करना, जैसा आपने अभी रैट के एग्जाम्पल में
डालने का।
बताया। हाँ, हेल्दी एनिमल में डालने का। फिर से क्या होगा,
हाँ।
डालने के बाद क्या होगा, डिसीज रिप्रोड्यूस इन द सेकंड एनिमल्स। ये एनिमल्स में
सेकेंड
क्या,
एनिमल।
हाँ, इसमें क्या होगा, फिर से
दूसरे एनिमल में वो डिसीज आ जाएगा।
आ जाएगा। ठीक है, तो माइक्रो ऑर्गेनिज्म और ग्रोथ आगे फिर क्या करने का, उसमें से आइसोलेट करने का, फिर उसको क्या करना है, प्योर कल्चर में ग्रोथ करने का।
प्योर कल्चर में।
ठीक है, फिर अगर समझे आइडेंटिफिकेशन ऑफ आइडेंटिकल माइक्रो ऑर्गेनिज्म, तो ये माइक्रो ऑर्गेनिज्म और ये माइक्रो ऑर्गेनिज्म अगर सेम है तो उससे क्या कंफर्म कर
हाँ,
सकते हैं वो, कि ये पर्टिकुलर डिसीज,
आइडेंटिकल माइक्रो।
हाँ, पर्टिकुलर डिसीज इससे हो गया। ठीक है, तो अपन ऐसा
यस
कंफर्म कर
सर।
सकते हैं। इतने स्टेप्स याद रखो, पोस्ट पोस्टुलेट क्या है ये याद रखो। ठीक है, इसमें कुछ पूछने का तो
यस
पूछ
सर।
सकते हो। हाँ, और दूसरी बात,
यस
सबको
सर।
सबको आवाज आ रही है, देखो अभी कौन कौन है
आ
ऑनलाइन।
रही सर, सबको आ रही।
हाँ, ये देखो, एक वो नाम लेके बोलता, अलीना सिद्दीकी। आवाज है कि, आवाज आ रही है अलीना।
आ रही सर।
जोया, जोया, आयशा,
ए जया।
आयशा,
आ रही सर, उनको भी, आयशा को भी।
तहसीन, तहसीन को आ रही है आवाज।
आ रही सर।
हाँ, तो खैर देखो अपने को क्या करने का, ये तो होगा थ्योरी।
लेट करंट बोलते
हाँ,
जाते।
थ्योरी का हो गया ये। हाँ, तो अब तो आपको एक प्रोजेक्ट देना है, आपको प्रोजेक्ट। हाँ, तो आप क्या करो इसमें, ये आज जो प्रेजेंट है, आज कौन कौन है, अलीना, अलीना यस बोलो,
हाँ। यस सर।
तहसीन,
यस सर।
जोया, आयशा, ये चारों क्या करने का, उन्होंने क्या, आप अपनी प्रोजेक्ट लेने का, एक सुन रहे क्या, प्रोजेक्ट क्या करने का है, इन्फेक्टेड वेजिटेबल्स कलेक्ट करना है। इन्फेक्टेड वेजिटेबल कलेक्ट करना है और उसके फोटोग्राफ्स लेना है। आप पहले इतना काम करो, इन्फेक्टेड वेजिटेबल्स और उसके इसके फोटोग्राफ्स करने का है। तो जितने घर में तरकारी
ठीक
लाते,
है सर।
जितने घर में तरकारी लाते,
हाँ।
इन्फेक्टेड उसकी फोटो लेने का, खाली खाली फोटो लेकर रखने का। हाँ,
ठीक है सर।
उसके बाद क्या करने का बताता हूँ। ठीक है,
ओके सर।
हाँ ठीक है, थैंक यू। फिर हाँ, आज
ओके सर।
स्टॉप करेंगे आज, और नेक्स्ट अपन दूसरे पीरियड में
यस
देखेंगे।
सर।
ठीक है, चलो, हाँ, अल्लाह हाफिज।
ओके सर। अल्लाह हाफिज।